So my lab at UCSF studies the tumor microenvironment in glioblastoma and its role in therapeutic resistance. And as a neurosurgeon, some of the things we sort of uniquely have access to is tissue, not just tissue, but sort of site-directed biopsies, regional tissue, as well as the ability to do intratumoral drug delivery. And I've been fortunate to be able to incorporate all of that into my laboratory practice to sort of make the surgeon-scientist model work in synergy. And that would be sort of the advice I would give if you're going to go down this pathway, would be to think about, I don't know why this is, sorry. So anyway, my lab at UCSF, I've been there for about 13 years now. And the way I would describe it is sort of that it's a 100-100 split, not a 50-50 split. But we work to molecularly define the components of the microenvironment using a variety of models and really a litany of people who've been with us over the years. So starting by who I am, I went to college at Stanford, did an MD PhD at Harvard, where my focus was on oncolytic viral therapies for glioblastoma. I then did my residency also on the East Coast, returned to the West Coast, as I said, in 2008 at UCSF, where I split my time between lab and clinic, although it really varies considerably from week to week. But at least at UCSF, the brain tumor labs are on a separate campus from a clinical practice. So in terms of juggling things, it's very important for me to be able to divide my time and in a manner that's sort of predictable and serves my trainees best. In the lab, though, there's a culture of fun and autonomy and teaching, as those who worked with me will attest to, which is exceedingly important because of how, that I'm juggling sort of these clinical duties, which creates an environment where people are not really thrive when they're more independent. We study the tumor microenvironment, which is really very important. It sort of evolved out of my initial work in oncolytic viral therapies and the recognition that tumors are not just masses of transformed malignant cells, but formed by the coevolution of cancer cells with the extracellular matrix, the tumor vessels, the immune cells, and become sort of these complete and dynamic organs. And so treatment strategies for glioblastoma must account for the dynamic interplay between tumor cells and their microenvironment, as shown in this schematic. And so some of the examples of things we've worked on, we've profiled tumor-associated macrophages in glioblastoma using single-cell sequencing, and really shown that this concept of pro and anti-tumoral macrophages is not a dichotomous distribution, but it's a spectrum rather than a dichotomous distribution. And we've been able to identify a number of targetable mediators and found that the process is indeed druggable. We've also done the same for tumor-associated neutrophils in glioblastoma and found that these similarly profile along pro and anti-tumoral fashions. And osteopontin is a targetable mediator of pro-tumoral effects of neutrophils that have emerged from that. And we've also looked at why therapies targeting the microenvironment have failed, including bevacizumab, which despite FDA approval has failed, and through a single-cell clonal profiling have found the transcriptional factor ZEB1 to be a targetable mediator of the response to bevacizumab. Similarly, for checkpoint inhibitors, we've identified that these have failed due to the T-cell depleted microenvironment of glioblastoma, and have done work to restore anti-tumoral T-cell function via retroviral delivery of immunomodulatory gene circuits. And some other projects we've been funded for is sort of building a 3D model of the tumor microenvironment and using it to screen for inhibitors of invasion. We're one of 12 national sites funded by the NCI to do work like this. And then lastly, we've been able to show that not just single molecules, but interactions between molecules are important for the pro-tumoral effects of interactions between cancer cells and the microenvironment by showing a structural complex between c-Met and beta-1 integrin that drives invasion. And this is all work that comes through tissue obtained from my operating room in sort of a site-directed manner where we're able to regionally profile the GBM microenvironment very thoroughly. And so that is really the 5-minute overview of everything we've done. none of which could be done without the numerous trainees, several of whom are on this call, as well as the funding sources that have made this possible. But really, if you want to learn more about the lab, and perhaps even more importantly for the people on this session, some advice for those of you who are considering sort of a surgeon scientist pathway, I'm very responsive to emails, particularly in this challenging applicant season. Happy to give advice anytime. And here's a photo of us, typically June is graduation time, and here's a photo of the lab from grad school graduation during a time when we didn't have to socially distance and sort of could stand next to each other, and hopefully that'll happen again soon. So thank you for your time this morning.
Hey everyone, Ryan Rad here from neurosurgerytraining.org. If you like that video, subscribe and donate to keep our content available for medical students across the world.